Hello everyone, this is Neeraj Pandey, Biofaculty. Students, as you know, at last day we have already discussed about a topic of cell biology. That's an overview of the cell. Now my child, today we are going to take another very important and wonderful session for the topic of cell biology, and that's the prokaryotic cell. Students, look at here. I am going to take a very interesting topic, that's the prokaryotic cell. Students, as you know, the examples of prokaryotic cells are the bacteria, mycoplasma, and cyanobacteria. Students, here I am going to discuss with the bacterium cell, the structure of a bacterium cell. See, this is the structure of bacterium cell. See. This is, I am going to mark here the boundaries of the bacterium cell. This is the boundaries of the bacterium cell. The outermost boundary, it is known as glycocalyx. And beneath the glycocalyx, another layer is present. It is known as cell wall, and beneath the cell wall, or you can say the innermost layer, this is the innermost layer, you can say this is the cell membrane. Okay, now collectively the outermost layer, glycocalyx, in middle cell wall, and innermost layer cell membrane. These three layers collectively known as cell envelope. Okay, now inside this cell membrane the fluid are filled. These fluids are filled. It is simply known as the fluid or matrix of the cell, and it is known as the cytoplasm. It is cytoplasm. Inside the cytoplasm there is genetic material, naked genetic material present here. I am going to mark here the genetic material of the cell. See, this is the DNA of the cell. This is the first strand of the DNA. I am going to mark the another strand, that means the second strand of the DNA. As you know, it is circular DNA and double stranded DNA. I am going to mark here, it is genetic material of the cell, that's the circular DNA. Circular DNA, but its circular DNA, it has the two strands, and that's why I am going to mark here DS, that means double stranded DNA. Okay, now the genetic material is circular double stranded DNA. Now it is single in number. It is single in number. Now this is the main circular DNA. Some extrachromosomal DNA, some circular extrachromosomal DNA are also present in some bacteria, like this. This is the extrachromosomal DNA present outside this circular DNA or genophore, and the name of this DNA is genophore. We can say these extra circular DNA present here, and it is named as plasmid. It is nothing but it is extra chromosomal DNA. Now these DNA, these plasmid DNA, they have certain phenotypic character. Later on we will discuss in the class 12 in the subject of biotechnology. Now see, in the cytoplasm, these particles, what I am marking here, it is a ribosome. It is ribosome. But you know, my child, the type of ribosome in prokaryotic cell, it is 70S type of ribosome. It is 70S type of ribosome. As you know, in the NCERT, the book that is written by God, that's the NCERT. In the NCERT, it is very clearly mentioned here, the ribosomes are attached. The ribosomes are attached with the cell membrane in prokaryotic cell like this. Okay, the ribosomes are attached with the cell membrane like this. When these ribosomes get polymerized with the help of mRNA. What I am marking here, this is the mRNA. The total structure, it is known as polysome or polyribosome. It is polysome or polyribosome. It is polysome or polyribosome. Now see, in the gram-positive bacteria, the inner side enfolding of cell membrane is present and it is named as mesosome. I am marking here another inner side enfolding of plasma membrane or cell membrane. See, this is inner side enfolding of cell membrane or plasma membrane. It is mesosome. It has various types of function. The main function of the mesosome is the respiration. The second, replication of the DNA and distribution in the daughter cell during the amitosis or binary fission. Okay. Now third function of this mesosome is the cell wall formation, etc. Now we are going to deal here another membranous structure present in the photosynthetic bacteriums, that's the chromatophore. These structures named as chromatophore. Chromatophore is nothing but the membranous structure in which the photosynthetic pigments are present, and that's why we can say the chromatophores are the analogous to the thylakoid of chloroplast. Okay, now see, we are going to discuss here some surface structures. We have three surface structures here. See, very small bristle-like structures are present here on the surface of bacteria. We can say this bristle-like structure is fimbriae. This is the fimbriae, and it makes attachment with the substratum. It helps to make attachment with the substratum. Now, another surface structure is present here, that is tubular in structure. Another surface structure is like this, tubular. Now, this tubular surface structure is known as pili, and this pili is composed a special type of protein, that's the pilin protein. It is what? Pilin protein. It is pilin protein. Now, another very important surface structure present that helps in the locomotion. Okay, now I am going to mark here another. See, this is the flagella, and flagella have three main structures. Flagella have three main structures. I am going to mark here. Okay. Now this yellowish part, it is named as basal body, and this reddish part, it is hook, and this long cylindrical and white-like part, thread-like part, it is filament. Now basal body, hook and filament, these three parts, they makes a locomotory part, that's the flagellum. It is flagellum. Okay, there are three surface structures. First, fimbriae. Second, pili. Pili helps in the conjugation, or you can say sort of sexual reproduction in bacteria. Secondly, flagella helps in the locomotion, and it is also composed very special type of protein, that's the flagellin protein. Flagellin protein. Okay, students, this is the very simple structure of a typical bacterium cell. Now, next day we will clear each and every status about this prokaryotic cell. Till then, tata.